Season 3. We'll be starting with the talks on molecular techniques, and the first talk of this out of the 3 talks that we'll be having on this topic will be molecular diagnostics of cancer, the solid tumors. The chairperson for today's talk is Dr. Kumar Prabash. He's a professor in the medical oncology department at TMC Mumbai. And the presenter of today's talk is Dr. Rakesh Demde. He's a molecular pathologist at Center of Oncopathology, Mumbai. The moderators of today's session are myself, Dr. Poonam Sharma, and Dr. Bonita Bora, who's a consultant pathologist at Kapili Nursing Home, Assam. And the webinar hosts are Dr. Anita Borges and Dr. Sumit Gujral. A few reminders. In case of connection interruption, low or no sound, video freezing or video lag, kindly leave the meeting and rejoin with the same link. And please do not send request for annotation to the presenters. These are few disclaimers. A few more disclaimers. And before we begin our first talk on the molecular techniques, that is the solid tumors, molecular diagnostics, a brief about the upcoming talks that we'll be having on the same, related to the same topics. On 21st November, that is next Tuesday, we'll be having the molecular diagnostics of cancer, hemato-oncology by Dr. Nikhil Patkar, who's professor in the Department of Hematopathology at ACTREC, Mumbai. And on 23rd November, we'll be having talk on cytogenetics and cancer diagnostics by Dr. Mayur Parihar, who's a consultant hematopathologist and molecular pathologist at TMC, Kolkata. So over to you, sir, Dr. Rakesh Demde.
Okay. I will just, you stop sharing, then I will share my screen.
Yes, sir.
So everyone can see my screen?
Yes, sir, it's visible.
So shall I start my talk?
Yes, sir.
Okay. So good evening, everyone. So today I will be speaking on basics in molecular diagnostic related to solid tumors. My whole talk will have broadly distributed in few basics terminology in molecular oncology. Then we will move to the hallmarks of cancer. We'll discuss something about the genetics of the hallmarks of cancer, how that is important to know in current molecular diagnostic technique. And then we will speak for a brief of what are the techniques we have at a molecular lab. So to begin, I will start with what is cancer. So cancer is a disease in which the body cell grows uncontrollably and spread to other parts of the body. And what is carcinoma? Carcinoma is basically cancer of an epithelial cell. So we have to know cancer is a broad terminology under which we have carcinomas, we have sarcomas, we have lymphomas and other type of cancer. So we will be focusing more of carcinoma in today's talk, and in the next talk you will be hearing about the hematolymphoid and other malignancies. Now, how does the cancer develops? So cancer is basically a genetic disease. So the genes in the body works in a harmony to for a proper functioning in a body. So whenever there is this harmony is disrupted either by some mutations or alterations, you get cancer. So basically cancer is associated with the molecular genetics. So majorly 3 types of genetic alterations are seen. One is in the proto-oncogene, the second one is a tumor suppressor gene, and third is a DNA repair gene. So in solid malignancy, these three are the more important pathways or genes affected in solid tumors. So in cancers, we can see the incidence of cancers in India is increasing. So this is a data of ICMR. So the data suggests that there is an increased incidence of carcinoma or the cancer in India predominantly due to the increased molecular technique to detect or to diagnose that particular cancer. And we can see from the same data, lung, mouth, prostate, tongue, and stomach are the more common cancer seen in male population. Breast, cervix, ovary, uterus, and lung are the predominant cancer types seen in the female population. So, with the increased incidence, it become very imperative to know about the genetics and the technique to diagnose as well as prognose these type of cancers in India. Before moving forward, we should know the basics of central dogma in molecular genetics. So the central dogma, what does it means? It means there is a DNA strand which transcribes to form one RNA. So this RNA undergoes poly A tail and it moves to the ribosome and it undergoes a translation to form a polypeptide chain which further convert into protein structure comprising of primary, secondary and tertiary. And this protein is the basic function of any major function we see in the body. So DNA have exon introns and that undergo transcription into a nuclear RNA. Then there is splicing scene where the introns are cut off and you get exon fusions and you get a messenger RNA and which gets converted into the protein, which is a functional unit. So in central dogma, what we have to know, the DNA, we see the mutations or copy number variations. For any fusions or any expression profiling, we generally look for the RNA or we look for some other epigenetic change which are not associated with the genes. And for protein studies, we have proteomics and transcriptomics. So this is the basics of central dogma of molecular genetics. So with this basic knowledge of transcription and translation, we move forward how the cancer is evolved. So in cancer evolution, we have two major type of mutations like driver mutations and passenger mutations. So as a name suggests, driver mutation, which provide a selective advantage and promote cancer development. So mutation which provide an advantage to cancer cell to proliferate and to invade and metastasis to other part of the body. Passenger mutations are the mutation which occurred in a cell as a coincidentally or subsequently acquired as a driver mutation. This passenger mutation doesn't provide any selective advantage. So we can see from this figure, the normal cells, you have a driver mutation and more additional driver mutation gets acquired by this particular cell and this particular cells proliferate and you get genetically heterogeneous cancer. So this is a basis of all the solid tumors. So in a solid tumors, you can get not a single but many driver mutation as well as passenger mutation. And in molecular genetics, we have to find out the driver mutations because that they are more important to be targeted than the passenger mutations. Then we have to know what are the type of genetic alterations. So we see mutations like point mutation. You have heard of point mutation, deletion mutation, gene rearrangement. So what are point mutation? So in a DNA sequence, there are nucleotide which are arranged. So whenever there is substitution of a normal nucleotide with another nucleotide and resulted in a conversion of that particular amino acid, you called it as point mutation. Or this kind of point mutation is commonly seen in an alteration like KRAS and TP53 gene. Then we have another deletion and insertion mutation. So this is an original sequence of nucleotide. So there will be mutation where a particular sequence of nucleotide is inserted in that and there is a mutation and the entire amino acid formation is changed. So this kind of mutation is called insertion mutation. Then we have deletion mutation. This is an original nucleotide sequence where the nucleotides are deleted and you get a something different amino acid. So these kind of mutations are commonly seen in KIT gene, RB1 gene and APC gene. Then we have another terminology called gene rearrangement and amplification. So you have heard about HER2 amplification in breast carcinoma. So what is happening? HER2 gene is a receptor which is present on the membrane of the cell. So HER2 amplification, there is a multiplication of this HER2 receptor occurs, and as a multiplication occurs, it dimerizes and it gives a signal for a further downstream cell proliferation. So this pathogenesis is commonly seen in the breast carcinoma. So this kind of alteration is called amplification. Similarly, you can get a gene rearrangement where you can see one part of ALK or one part of gene fuses with another part of gene. So we have ALK gene which fuses with EML4 gene, which is commonly seen in lung adenocarcinoma, and this activates the further downstream pathway, and you get a lung adenocarcinoma. So this kind of alteration is called gene arrangement or gene fusion. So you have to, whenever you see any mutation, you have to check whether it is point mutation, deletion, insertion mutation, or gene arrangement. Because in gene arrangement, you have to go and look for an RNA based studies, and in mutation, you have to look for alteration in a DNA strand. In molecular, you can also see large cell transition. So this is a chromosome which have two alleles, and both the alleles have one maternal and one paternal. So the heterozygosity is maintained. Whenever some alteration which is large scale and causes insertion or deletion of one allele, you get loss of heterozygosity. So loss of heterozygosity is also an hallmark of cancer, which is seen in solid tumor. Similarly, you can get other like copy neutral loss of heterozygosity, where there is a change in the both the alleles, but the copy numbers remain same. So such type of large scale transition is also seen in solid tumor. So molecular genetic techniques are there to diagnose such kind of transition, but we have cytogenetics which deals with such large scale transition. MLPA is one molecular technique where we can see such copy number variations also. But we are more focused about the small transitions than the large scale transition when we talk about the molecular diagnostic technique in solid tumor. We move to what is the utility. After we seen some of the basics of what are the type of mutation, what are the type of alteration, why we should go for molecular pathology in cancer or molecular diagnostic. So we can use molecular pathology for diagnosis of a particular cancer. For example, we have BRAF V600E mutation, which is a diagnostic in melanoma. We can also have fusion between EWSR1 and FLY1 gene, which is a diagnostic of Ewing sarcoma. Then we have some markers which are prognostic to a particular solid tumors, like TP53 mutation in colorectal have a poor prognosis, and it is also associated with the hereditary association of colorectal cancer. Similarly, HER2 gene amplification also has a bad prognosis. So so the molecular technique can guide us about the prognosis of a particular solid tumor. Similarly, we can use this molecular diagnostic technique for therapeutic approaches also. For example, EGFR mutation in lung adenocarcinoma can be targeted by the tyrosine kinase inhibitor. So we can get this EGFR mutation and we can give a treatment of tyrosine kinase inhibitor like EGFR inhibitors, first generation, second generation, depending on the type of mutation in EGFR gene. Similarly, in KIT mutation in GIST also can be targeted. We have to know what type of exon is mutated. We can give first generation, second generation, and third generation of TKI in this kind of GIST malignancy. So molecular pathology can be utilized in all the three parameters. Now we will discuss something about the hallmark of cancer. So there are 6 hallmarks which we all know, and there are emerging hallmarks like genome instability, tumor promoting inflammation, and hallmark associated with energy metabolism, and the hallmark associated how the immune destruction can be evaded. So these hallmarks of cancers are also associated with the molecular alteration. So cancer cell try to mutate in such a way that this all the 6 parameters and the new emerging parameters can be achieved by a particular cancer cell. For example, sustaining proliferative signaling. So the mutation will occur in a cancer cell so that the proliferation is uncontrolled. So there can be examples which I will be discussing in brief in the slides. Then we have evading growth suppressor. So there is a cancer cell and there is a tumor suppressor gene who tries to control the growth of a particular cell. So there will be mutation which is occurring in a particular cell which will evade the suppressive effect of a particular gene. So you can see the mutation which try to evade this growth suppressor as a hallmark of cancer, and cells try to evade most of the growth suppressor, try to mutate. There can be gene mutation which can be seen, which which evades this growth suppressor. Then there are act then the cancer cell always try to invade and metastasize. So there can be mutation which can be seen, which activate the invasion, or which increases the invasive ability of a particular cancer cells. So such type of hallmark are associated with invasion and metastasis. Then cancer cell, with the help of some mutations, try to enable the replicative immortality. The one of the gene is a third promoter mutation, or telomerase reactivation is one of the example of enabling replicative immortality. Then cancer cells also require a good circulation or nutrition for the growth. So the cancer cell will release the biomolecules, so that there will be proliferation of the blood vessels surrounding the cancer cell, and which will provide the nutrients to a particular cancer, so that they can grow in size. Then the cancer cells also have mutations which can resist the cell death, like TP53 mutation and MDM2 amplification. So these all hallmarks or the genetic pathways we will discuss one by one in the slides. And we also will discuss what are the emerging hallmarks like genome instability. We will discuss about the energy metabolism, and we will discuss tumor promoting inflammation in my next slides. So the first one is signaling pathway, which carries a very important part of a molecular pathology in cancer development. So basic stages in cell signaling is that a signal molecules arrives at a target cell. So this is basically a receptor. A signal molecules or the growth factor comes to a receptor. It attaches to a receptor, and on attachment there is some phosphorylation which takes place. An inactive signal transduction molecule is converted into active transduction molecule, and this active transduction molecules further activates the downstream pathway and goes to the nucleus and activate the transcription. So this is the basic mechanism how that signal transduction pathway takes place. What is the importance of knowing the signaling pathway? Because of this signal transduction pathway, there is a continuous proliferation of cell. The cell undergo differentiation from epithelial to mesenchymal. There is a lot of protein synthesis takes place, which helps in cell growth, and the cell can use the signaling pathway for angiogenesis as well as invasion and metastasis. So the signal transduction pathway uh, has an important role in all the major hallmarks of cancer which we have discussed: proliferation, angiogenesis, adhesion and metastasis, and inhibition of apoptosis by TP53 or other pathways. In cell signaling pathway, there are two types of tyrosine kinase activity what we see. So tyrosine kinase is basically involved receptor type of tyrosine kinase activity and non-receptor. So receptor type of tyrosine kinase have a receptor which is bound to the plasma membrane, which have a area where a growth factor or ligand binds. On binding, it further downstream phosphorylate the other transaction pathway, which can be a non-urticate type of tyrosine kinase. For example, EGFR is a receptor, so ligand can bind to EGFR. Phosphorylation further downstream activate the RAS and RAF pathway, which can be our non-receptor, which are not attached to the receptor, but will have a tyrosine kinase, intrinsic tyrosine kinase activity. And this signal transduction pathway will further help cell to proliferate and evade cell proliferation, as well as the other important like angiogenesis, and evade the apoptosis. So these are the 10 signaling pathway which is demonstrated by TCG, or which is mentioned in the cancer genome atlas, which are having an importance in solid tumor pathogenesis. So we have RTK-RAS, PI3K, AKT, mTOR pathway, which are the two major pathway which is seen in majority of the solid tumors. Then we have WT beta catenin, TGF beta, SMAD pathway, which is seen in colorectal or some other hepatocellular carcinomas. and some other notch signaling and hyposignaling, and p53 pathway, which is a major pathway for apoptosis. So we'll discuss few of the pathways among these 10, and we see what are the alteration occurring in this pathway and which malignancies are seen in particular alterations. So first pathway is RTK, which is very important pathway because it has seen in majority of the solid malignancies, even in the hematological malignancies also, we can see the alteration of this pathway. So this pathway get activated of by numerous receptor which are present on the membrane. So we have receptor like EGFR, which is a RTK receptor. We have HER2, which is present on the plasma membrane, and we have fibroblast growth factor like receptor also. KIT, MET, and insulin growth factor receptor. So this all receptor downstream activate the KRAS, HRAS or NRAS, and which further downstream activate the BRAF and the downstream signaling in the nuclear transcription factor activation, and you get cell proliferation, cell survival, and translation to a protein which helps in the angiogenesis and cell survival. So this is a basic signal transaction pathway we see in RTK RAS pathway. This pathway is very important because you see in colorectal cancer, you have RTK-RAS pathway alteration. You have lung adenocarcinoma also, RTK-RAS pathway, breast, urothelial, and melanoma. So these are the major carcinoma which we have seen in the incidence. These are the carcinomas we see a majority in the Indian population, and the pathway which is altered is RTK-RAS. So we should have a knowledge of this pathway because there are many biomarkers or targeted therapy which is FDA approved for particular malignancy associated with RTK and RAS pathway. So these are the FDA approved targeted therapies which are available against the RTK RAS. We have EGFR inhibitors which we give in lung adenocarcinoma. ALK rearrangement, we have ALK inhibitors in lung adenocarcinoma. We have ROS also FDA approved. Crizotinib is there available for lung adenocarcinoma. In HER2 amplification, we have trastuzumab and other generation of HER2 inhibitors. We have MET. So whenever we have MET amplification or MET mutation, we have a FDA approved crizotinib. This can be seen in lung adenocarcinoma. This is also an FDA approved MET inhibitors. So these are the FDA approved because this is a major pathway in most of the solid tumors. So you have a lot of FDA approved targeted therapy in RTK RAS pathway in solid malignancies. Now we move to the other proliferative signaling alteration pathway, which is APC WNT beta catenin pathway. So this pathway is altered in colon cancer, predominantly in FAP type of colon carcinomas. Then you have hepatocellular adenoma also, where you can see WNT beta catenin pathway alteration. So this APC WNT beta catenin is basically a tumor suppressor pathway where you have result shaped WNT receptor present on the plasma membrane, and you have a complex which have APC, axin, and other GSK and CK1 molecules. So what this molecule do? This will cause a proteasomal degradation of beta catenin in the cytosol. So whenever there is no ligand attachment to particular WNT pathway, this will cause a degradation of beta catenin, and once the beta catenin level is low in cytosol, there will be no transcription and no cell proliferation. So this is a basic basic mechanism of this WNT beta catenin pathway. Whenever a WNT ligand gets bind to this receptor, the beta catenin is released. There is no proteasomal degradation, and this beta catenin moves to the nucleus. It binds to the transcription factor, and you can get transcription and further proliferation of a cell. So whenever there is alteration of this pathway, you have beta catenin gene like CTNNB1 gene, which encodes this beta catenin, and that can be altered in colorectal cancers, as well as APC gene. We know that pathogenesis of PAP, the APC gene is the first molecular alterations in colon carcinoma. So both these genes are associated with WNT beta catenin pathway. So we should know about, whenever we are dealing with colorectal cancer, we should know where this pathway and what is the importance of this pathway, because this is a tumor suppressor, and whenever there is alteration, this tumor suppressive effect is gone and you get lot of proliferation. The next is a MYC pathway. So all the receptor which are present on the plasma membrane are RTK receptor. They combine and comes to the nucleus, and in nucleus there are cyclin dependent kinases present, and there are cyclin available. So this cyclin and cyclin dependent kinases will cause the proliferation or will activate the further transcription of particular gene. So MYC is one of the gene which goes and bind with MAX. In a normal physiology, this will lead to the proliferation, this transcription of a particular DNA segment, and this transcription further lead to a translation into protein, and there will be a proliferation of a particular cell occur. So whenever there is MYC pathway altered, means there is a MYC amplification or MYC overexpression. So the MYC is present in large quantity, it will go and bind with lot of MAX, and there will be lot of transcription, and there is lot of cell proliferation seen. So there can be a normal RTK functioning, but this MYC can be amplified, or the numbers increases, or the expression of MYC is increased. In that case also you get a lot of proliferation. The common malignancies which gets affected, which is involved by this MYC pathways, are Burkitt lymphoma. In solid tumors, you have small cell lung carcinomas and neuroplastomas. The other areas like cervical, colon, and breast, still MYC pathway role is under trial. We move to the next hallmark, which is evading growth suppression. So we have here another RTK or signaling pathway, which is PI3K, AKT, mTOR pathway, and you have P10. So P10 is a molecule which suppresses or which put a brake on this PI3K pathway by preventing this phosphorylation of PI3K. Okay, so whenever there is alteration of P10 gene, which can be seen in examples like glioblastoma, up to 50%, endometrial carcinoma 35%, in prostate carcinoma 40 to 50%. Whenever this P10 get mutated, there is no break, and the PI3K AKT mTOR pathway is activated, and this is a proliferative signaling pathway, and you get lot of proliferation signal, and it further translate into a tumorigenesis. So P10 is seen in solid tumors, and we have examples where it is seen predominantly. In endometrial carcinoma, we also have some P10 methylation, which is an epigenetic changes, which we'll discuss further. Apart from mutation, if the promoter region of P10 is also methylated, the functioning of P10 will not take place, and there is a loss of this suppressive effect. The next hallmark or the next pathway is TGFR beta receptor, or transforming growth factor receptor pathway. So these have a dual role. So TGF beta is basically a tumor suppressive pathway, where it tries to suppress the signal for proliferation by downstream activation of SMAD2 and 3, which goes and binds SMAD4, and which will prevent the transcription factor for its functioning. So this is a role of tumor suppression. Then it also have a role where it modify the inflammatory signals or inflammatory cell function. So TGF beta also have a role in inflammatory signal or inflammatory tumorigenesis. This is one of the normal function of TGF beta. So whenever TGF beta is mutated, so you get a lot of downstream signaling, because this break of SMAD complex is gone, and you have a lot of transcription factor. So you get a lot of signaling where the cell proliferation start, as well as the mutated TGF beta also modulate the inflammatory environment of a particular tumor cell. So you get lot of tumor proliferative added advantage from this inflammatory cells. So this pathway have the role of proliferation as well as inflammation. The second important role in of invasion and metastasis is also done by TGF beta. So TGF beta is primarily involved in epithelial mesenchymal transition. So what does it mean by epithelial mesenchymal transition? You have normal epithelium, it is very much compactly packed, and there is adhesion scenes between both the cell. So whenever cancer cell grows, there is loss of adhesion. The one gene which is involved in the loss of adhesion is cadherin gene. The other, like fat1, is also involved, where the adhesion is lost and cell try to get separated from each other. So this is one of the hallmark of cancer cell, where the cell try to get separated from each other. The other thing, what happens whenever there is TGF beta mutation, the second thing what is happened, this normal cancer cell, which is epithelial growth, convert into a mesenchymal or more toward the stromal component or stromal cell looking of a particular cancer cell occur. So what does happen whenever there is a mesenchymal component acquired by this cancer cell, it becomes more resistant to the targeted therapy, and it becomes gains a power so that it can invade further and or to a distant metastasis. So whenever you have TGF beta mutation, you can see a high grade carcinomas where you can see mesenchymal type of cancer cells, and it is seen in colorectal cancers, pancreatic cancers, breast cancers, and endometrial cancer. So TGF beta pathway has a role in tumor suppressions as well as a invasions and metastasis, where it converts a particular cancer epithelial cell into mesenchymal cell. Because once the cell becomes mesenchymal, it becomes very resistant to the normal targeted therapies or chemotherapies, and it gains the power to invade to a distance organs or the other organs. So TGF beta is seen in high grade carcinomas like pancreatic cancer. So TGF beta is one of the uh, more important pathway to know whenever we speak of a high grade carcinomas or high grade solid tumors. The next hallmark is angiogenesis. So whenever a cancer cell wants to grow or to grow in a large cells, they require blood supply, they require the nutrients to grow. So to grow that, you have the endothelial cells. So these endothelial cells has to undergo proliferation to produce a new vessel. So this endothelial cell proliferation is taken in a high number because of some other biomolecules. So cancer cell release biomolecules like vascular endothelial growth factor. So this is one of the biomolecule which will go and attach to the endothelial cell receptor, and which gives signals to this endothelial cells to proliferate. And apart from this cancer cell, there are tumor inflammatory cells which is present in a cancer cell surrounding. This also releases vascular endothelial growth factor, and it causes the proliferation of these endothelial cells. So the inflammatory cells try to provide a good microenvironment for a cancer cell is also known in recent times, where they release vascular endothelial growth factor, as well as the macrophages can release platelet derived growth factor, which also act on these endothelial cells, and you get a lot of proliferated endothelial cells, and you get a lot of angiogenesis in a particular carcinoma. There is cancer associated fibroblast also, which also release fibroblast growth factor, which also act on this endothelial cell, and this all particular biomolecules come and act on their individual receptor, like vascular endothelial growth factor receptor 1 and receptor 2 is active, where the biomolecule VEGF comes and join, and further downstream transduction pathway is activated, and there is a proliferation of particular endothelial cell. Similarly, you have PDGFR receptor, where PDGFR molecule comes and join. So that this is how a particular angiogenesis is favored, not only by the cancer cell but also from the inflammatory cells and other fibroblasts which is present in the microenvironment of a particular solid tumor. Now, based on whatever I have spoken till now, there is one question which the audience has to give the answer. So, what is the signal transaction pathway in cancer cell leads to all? So, there are signal transaction pathway which we have discussed in the previous slide, except what are the benefit they give to the cancer cells. So the option 1 is cell proliferation, the option B is invasion and metastasis, the option C is apoptosis, and option D is angiogenesis. You have to select except. What are the benefit or what are the advantages cancer cell gets from signal transduction pathway? Except you have to answer.
Sir, majority of the participants are answering the option C, followed by B.
Okay. So signal transaction pathway, the answer is C. So signal transaction pathway C, the cancer cell will never try to increase the apoptosis, will try to inhibit this apoptosis. No? The cancer cell doesn't want the cancer cell to die. Okay, so this is the odd or this is the not the thing which the cancer cell will do with the help of signal transaction pathway. Cell proliferation, definitely there will be alteration which will lead to the cell proliferation. There will be a transaction pathway which we have already discussed, TGF beta, where leads to invasion and metastasis. Angiogenesis, we have seen again the signal transaction pathway from the receptor to the downstream, you get the proliferation of endothelial cell. So apoptosis is the one component which the cancer cell try to avoid. So apoptosis inhibition is one of the step which signal transaction pathway will do, not the apoptosis. So answer is C. The next hallmark is resisting cell death or genomic stability. So we all know P53 is a guardian of genome. So whenever there is a DNA damage occurs, so TP53 accumulates in that around in that cell, and it will try to arrest that cell cycle. It will give time to the cell for a repair of a particular DNA. Once the DNA repair is good, then it will lead to further cell cycle from whatever step it has arrested. Okay, but whenever the DNA repair failure occurs, so it will lead to the apoptosis of a particular cell or death of a particular cell. So P53 is a what you call is a control of that apoptosis or cell death. Then what is this MDM2? MDM2 is a gene which controls this P53. Okay, it will cause degradation of P53. So basically, it is a break of P53 functioning. So whenever either P53 get mutated or MDM2 get more in number, you get less TP53 and the cancer cell will try to avoid this apoptosis. Okay, so you can either get P53 mutation or you can get more number of MDM2 which will prevent break on P53 functioning. So we have an example where TP53 mutations TP53 mutation is seen in a lot of malignancies, but we have ovarian carcinoma, colorectal, lung, breast, and the hereditary or germline mutation of TP53 life syndrome is also associated with many type of solid tumors. Similarly, MDM2 amplification or the number where the MDM2 is increased and will prevent the functioning or degradation of P53 increase, you see in sarcomas. Okay, myxoid liposarcoma example. We can also see MDM2 amplification in some of the breast carcinomas and other malignancies. So this is how the cell death has been resisted by a particular cancer cell. Till now, we have discussed the genetic part where the mutation, alteration, deletions occurs in a cancer. Now there is something called epigenetic. Okay, so epigenetic is basically a study of expression of gene. Okay, and this particular expression of whatever component of epigenetic is there, it is heritable. So whenever a cell proliferate, this particular epigenetic change is also transferred to the progeny. Okay, so the epigenetic doesn't alter with the cell division. Okay, so this is a heritable study of alteration in gene expression. So we can see whenever there is a DNA who has to undergo transcription, before that RNA polymerase has to come and bind to a promoter region of a DNA so that DNA get activated and it undergoes transcription to form the mRNA. But this DNA is not so easily available because this DNA is coiled around a histone protein, because DNA is very long, and to accommodate that much DNA strand, it is coiled around the histone proteins. So octamer of histone is called a nucleo. Whenever there is 8 histone comes together, it is called nucleosome. So these are very compactly packed, and there is no way that the DNA strand gets this transcription factor like RNA polymerase go and bind whenever they are in heterochromatin or compact state. So this has to be unwind or separated so that the DNA strand of interest gets exposed and this RNA polymerase go and bind to a particular DNA strand. So this is a histone protein. We have H2A, H2B, H3, H4, four types of histone proteins around which the DNA molecules just wrap. So they have also tail around particular histone. So whenever histone has to unrelease or get loosed, this particular tail has to undergo acetylation. Acetylation is a process where acetyl group is added and it undergoes release of a particular DNA. Okay, so this is a very simple way of explaining the epigenetic, but it is a very complex process where there are a lot of things which happens apart from acetylation. There is one pathway called HWI-SNF pathway, which is also called switch pathway, which goes and bind to particular region of this histone, and it will just separate two histones and expose the particular DNA strand. So whatever I am telling is very simplistic, but it is a very complex process. There are many things which is involved in epigenetic of a particular gene. So why we want to know this epigenetic change in solid tumors? Because there are two processes which takes place in epigenetic. One is methylation and another is acetylation. Whenever methylation means whenever a methyl group goes and bind to a promoter region like CpG island promoter region goes and bind to cytosine group. Okay, it will silence that particular gene because the promoter region is silenced due to this methylation. Okay, so in solid tumor, whenever there is a methylation of a particular tumor suppressor, the function of tumor suppressor will not happen and will lead to tumorigenesis. So we should know the methylation status of a tumor suppressor, and we should know the acetylation which will increase the transcription or which will give the order to promoter region to further go and transcribe to a particular RNA. So we should know the acetylation of a particular oncogene or proto-oncogene, and we should also know the methylation status of a promoter region of tumor suppressor gene. So these two basic concepts you have to know whenever you deal with the epigenetic. So I will give you example. There is pancreatic ductal carcinoma where epigenetic regulator. So this ARID1 and SMARCA2 are the two genes which I have told you are a part of switch pathway. So this gene get altered, so there will be a lot of opening of this particular or unwinding of this DNA strand occur. Okay, and the transcription factor will come and bind with this particular opened or unwinded DNA strand. So there will be a lot of transcription or in downstream there will be a lot of proliferation. Okay, so you can get epigenetic regulator mutation in pancreatic ductal carcinoma of switch pathway. Then there can be hypermethylation. As I've told you, methylation silences a particular gene. Okay, so TGF beta, we know it has a tumor suppressive activity. Similar WNT beta catenin also has a tumor suppressive effect. So whenever methylation occurs of this particular gene, the tumor suppressive effect is gone and the cell is free to proliferate. So these are the epigenetic changes also seen in solid tumor. When then we come to the next example, triple negative breast carcinoma. So we have BRCA1. Okay, so BRCA1 promoter region is hypermethylated. So BRCA1 is a gene which has a role in DNA repair pathway, which we'll discuss later part of this lecture. So whenever the hypermethylation, as I told you, methylation silences a gene. So the DNA repair will not take place and genome become very highly unstable. And this high unstable genome will causes the proliferation or mutation in a progeny of cells which we get in breast carcinoma. So this is commonly seen in triple negative breast carcinoma where ER, PR and HER2 all three are negative. Then we have a role in therapeutic approach where we check the methylation status of MGMT in glioblastoma multiforme. So this hypermethylation status of MGMT we check for giving a therapeutic drug. So epigenetic alterations also has role diagnostic as well as therapeutic. Now we will, after discussing the genetic and epigenetic alteration, now we will discuss in brief the more commonly occurring malignancies which we encountered in molecular lab, and we will also discuss the malignancies or cancers which we have a lot of biomarkers or targeted therapy. The first one is a non-small cell lung cancer. So lung cancer we have broadly adenocarcinoma, squamous, and other variant. And in adenocarcinoma, with the advancement of molecular genetic techniques, we can see mutations in KRAS, EGFR, ALK, ROS. These are all part of our RTK-RAS pathway. Then we have MET, PIK3CA, MAP2K kinase, which is again a part of PIK3CA mTOR pathway. So majority of the alteration in adenocarcinoma lung take place in the RTK-RAS or PI3K MAP2 kinase pathway, which are the two proliferative signaling pathway of the hallmark of cancer, are commonly involved in adenocarcinoma. When we move to the squamous carcinoma, again we have PIK3CA, which is a proliferative signaling pathway. We have alteration in PTEN also, which we have already discussed. There is a loss of PTEN, and then which is a break for PIK3CA mTOR pathway, which we see in squamous cell carcinoma. So these are the molecular alteration which takes place in adeno as well as squamous carcinoma. And why we have to know the knowledge, because there are a lot of targeted therapy in non-small cell lung carcinoma against all this alteration available, which is FDA approved, and we have to give the mutation status so that the targeted therapy can be given to the patients. The most important gene which get mutated in non-small cell lung carcinoma or adenocarcinoma are EGFR gene. So in EGFR gene also, the tyrosine kinase domain, which comprises of exon 18, 19, 20, 21, are commonly affected, of which exon 19 and 21 are affected in more than 90% of the lung adenocarcinoma. Okay, then why we have to know this particular deletion, exon 19 and exon 21, specifically mutation of exon 19? Because when you get exon 19 and exon 21 mutation, you can give first generation TKI or tyrosine kinase inhibitor. But there are some mutations which uh, acquired as a secondary mutation in a particular clone of cancer cell in lung adenocarcinoma. One of the mutation is T790M, which is a point mutation of exon 20. Okay, whenever we get this type of mutation, the first and second generation of tyrosine kinase inhibitor remain ineffective, and you have to give a higher generation, like osimertinib is a treatment of choice for such kind of mutation. So in EGFR mutation, we don't have to just tell ki this is EGFR mutation. We have to tell specifically which exon, which type of codon or which type of mutation is affected in EGFR. Because apart from T790M, there are other resistant mutations also to tyrosine kinase inhibitor. One is, we have already discussed, T790M. There can be insertion type of mutation in exon 20 also occurring. There are other point mutation or missense mutation occurs in various codon, 768, 747, 761. So we, in molecular diagnostic, in specifically in lung adenocarcinoma, we have to specify what codon and what exon mutation is there, because the drug is available to treat such kind of mutation. Apart from EGFR mutation, where the TKI doesn't work, you can get mutation in other RTK like KRAS, BRAF, insulin like growth factor mutation, and MET amplification. So these are also an other resistant mutation which the lung adenocarcinoma cell acquire, and your EGFR tyrosine kinase inhibitor doesn't work. The next, after EGFR, we see a lot of ALK gene rearrangement in lung adenocarcinoma. So the routine practice of ALK rearrangement in routine, or the more commonly practice area, is by IHC. So we get a lot of tests for EGFR mutation and ALK and ROS IHC. But with this example, I want to highlight, in ALK also we have to be very specific about what is the type of fusion we are dealing with. Okay, so there are two genes, EML4 and ALK. Both are present on chromosome 2. So there is a breakage of EML4 occurs. It gets it changes its direction and fuses with the ALK. So you have EML4 and ALK fusion, and this particular fusion will activate the downstream signaling pathway and lead to the proliferation of a adenocarcinoma cell. So now there is a classification or sub classification of these particular fusions, V1, V2, V3, and there are other further downstream classification. But we have to be very specific about these three sub classification of EML4 ALK, V1, V2, and V3, where V1 there is exon 13 of EML4 where it get breaks and it fuses with the exon 20 of ALK. Similarly, 2020, exon 20 and exon 20 of both EML and ALK get fused. In V3 variant, you have exon 6 of EML4 and exon 20 of ALK. So knowing this V3 type of fusion is important because it is having a poor prognosis and it is resistant to the first generation of ALK inhibitor. So in molecular, we can get this sub classified sub level of information which we cannot get from IHC of ALK and ROS. And we can know ki V1 is the predominant type of fusion which we get in non-small cell lung carcinoma, followed by V2 and V3. So whenever we get V3 type of fusion, it is having a poor prognosis and there is a resistance to the first generation TKI. So essential biomarker earlier and which is also predominantly done in many centers are EGFR, ALK, ROS, BRAF, and PDL1, which is done by IHC. So biomarker like EGFR can be done by the simple technique PCR, real-time PCR is routinely used. Then you can go for NGS, ALK, ROS by IHC is commonly done. It can be done by FISH, where we can see the rearrangement or break apart probe, where we can know there is a fusion or rearrangement occurs with the ALK, but we don't know what is the partner where the ALK has gone and combined with. In NGS, we can get all the information of EGFR, ALK, ROS, BRAF in a single assay. PDL1 has to be done by IHC. So in lung carcinomas, we also face this challenge of tissue is the issue, where you get a lot of core biopsies, and when you go and do individual biomarker testing for ALK, ROS, EGFR, you tend to deplete the tissue, and it becomes very difficult whenever you have to get information out of like information of the other genes which are not present in our regular biomarker testing. So you have to you don't have tissue to go for a next generation sequencing panels. So in such cases, the strategies or as the test of next generation sequencing becomes a little bit cheaper in lung carcinomas, you can directly order for next generation sequencing where the tissue can be preserved, or there can be two course of biopsies can be done, one for ISCs and the other for the next generation sequencing, where we can detect apart from this regular ALK, ROS and EGFR the other biomarkers which causes resistance to the treatment or which have a prognostic impact. So lung carcinomas or lung non-small cell carcinomas have a lot of targeted therapy against EGFR, ALK, ROS. There is a BRAF also seen in lung adenocarcinomas. We have a treatment. We have NTRK, which is one of the type of same tyrosine kinase receptor present. So there are NTRK 1, 2, 3. You have a targeted therapy, and it is also seen in non-small cell lung carcinoma, specifically in the adenocarcinoma. You have red positive cases. You have MET exon 14 skipping mutations seen. So you have a lot of uh, emerging biomarkers or targeted genes in lung adenocarcinoma. So sticking to the older strategy of EGFR ALK ROS will not be sufficient for advanced non-small cell lung carcinoma or adenocarcinoma. You have to get an information or comprehensive information of entirety for lung carcinomas, and there is a lot of tissue issue depletion of tissues also occurring in lung carcinoma. So I think we should go for upfront NGS in cases of lung adenocarcinoma. We move to the other important colorectal carcinoma pathway. So in colorectal carcinoma pathway, we have two pathways. First is the APC beta catenin, which start with FAP. You have a mutation in APC gene, and you get hyperproliferative epithelium, which convert into adenoma. Then the further mutations are acquired in KRAS gene, SMART2 and 4 of TGF beta and TP53, and it leads to the adenocarcinoma, grown adenocarcinoma. And due to the additional mutation acquired, this particular cancer cell try to metastasize to a distant site, like invasions and distant metastasis can be seen because of the acquire of this additional mutation. Then there is another pathway called microsatellite instability pathway, which is associated with the HNPCC. And in this pathway, the microsatellite become unstable, or there is instability occurring in the microsatellite. So microsatellite is basically it is a 1 to 6 nucleotide repeat which occurs in tandem. Tandem is one after the other. So there can be a repeat in nucleotide like CAG, it repeating CAG, CAG, CAG. So this is three nucleotide which get repeat. So if their nucleotide repeat is around 6, then you have a microsatellite. So whenever microsatellite instability occurs, you get an HNPCC type of colorectal carcinomas. And the genes which are involved are mismatch repair genes like MLH1, MSH2, MSH6, PMS2. You get mutations in this particular genes, and these genes have a hereditary connections are seen in these genes also, in the form of Lynch syndromes. So these genes, apart from colorectal cancers, we can also see in endometrial carcinomas, also few of the gastric carcinomas also you can get a mutation of this mismatch repair genes. So this instability leads to a formation of adenoma, and further this adenoma acquire the other mutations of signal transduction pathway or anti-apoptotic pathway like BAX or TGF beta, and you get a full-blown adenocarcinoma and tumor metastasis. So this is molecular genetics associated with, in brief, about colorectal carcinomas. This is a published study in 2015 where they have taken into consideration 6 different subsets of classification of colorectal cancers, and they have combined and came to a consensus molecular subtyping into 4 categories. We have CMS1, CMS2, CMS3, and CMS4. So in the first consensus molecular typing, it is more associated with MSI or micro satellite instability. So you get whenever you get microsatellite instability and you have BRAF mutation, so you have the colorectal cancers in this category. There is a lot of immune infiltration, and in recent times, immunotherapy can be given as MSI is very high. Then you have the second molecular subtyping where WNT and MYC activation is seen, which is seen in like APC WNT pathway is commonly involved. So this is around 37% of our colorectal cancers. We have third category where metabolic pathways are generally altered, and along with metabolic pathway, we have KRAS mutations. So whenever KRAS mutations and metabolic pathways is affected, you can subclassify the particular colorectal cancer in this category. We have the last mesenchymal, as I have already told, this TGF beta activation leads to mesenchymal formation of a particular epithelial carcinoma, and it is having worse relapse-free survival. In category 1, whenever there is a relapse, it will have a worse survival. The other two categories, the survival has not been defined. The next solid tumors where the molecular diagnostic has a role is gastrointestinal stromal tumor. So this is a stromal tumor which affect the stomach, the duodenum and jejunum. It is diagnosed by an IHC called KIT and DOG1. So this IHC are used to diagnose gastrointestinal stromal tumor. So just diagnosing will not suffice, because the molecular alteration in which exons of our KIT gene is affected or PDGFR gene is affected, we have to know. So we can see in this figure, exon 11 is the common or predominantly affected KIT exon, followed by exon 9, and there are secondary acquired mutation which we can see in exon 13 and exon 17. So exon 9 and 11 you can see on this flow chart. Whenever there is exon 11 mutation, we can give imatinib. Whenever exon 9, we can try with higher dose of imatinib. And if you have other mutation like exon 13 and exon 17, you have to go for a higher generation of TKI. So knowing the type of mutation, type of exon, the treatment changes in GIST. Similarly, goes with the PDGFR mutation. Exon 18, specifically codon 842, are commonly involved. So if you have 842 mutation, you can give a drug, PDGFR inhibitor. If it is other than 842 codon mutation, you can stick with the imatinib. So knowing a particular mutation has a major therapeutic role in GIST. And in GIST, just KIT mutation, KIT, IHC and DOG1 will not suffice with the treatment part of GIST. Now the second question which I will ask to the audience: most common gene mutated in lung adenocarcinoma. The option is KIT, B option is EGFR, third is MLH1, and D is ATM.
So should I tell the answers in the chat?
Yeah,
So
yeah.
majority, all the participants have answered the option B.
Okay. So the common gene mutated in lung adenocarcinoma EGFR, of which we have seen exon 18 deletion and missense mutation of exon 20, that is L858R, are 90% of the mutated EGFR in lung adenocarcinoma. We move to the next hallmark, which is the which enable the replicative immortality. So the telomere are present on both the ends of our chromosome, and which with each cell division, a particular region of telomere get shortened. So there will be time after 50 to 70 cell division, the telomere will be lost and cell cannot go further for a proliferation, and such cells undergo a p53 driven apoptosis. But there can be sometime repair of a particular strand of a DNA or chromosome, which is called non-homologous end joining, where a particular end of one chromosome get attached to the other chromosome, like you can see in this picture, and it becomes dicentric chromosome. It undergoes further cell cycle, and you can get a new double stranded break. And there will be a time comes when this doesn't the cell loses the capacity to repair this particular new type of double stranded break, so it will again go to cell death. This happens because the telomere length cannot be maintained in a cell. So this can be maintained whenever there is a reactivation of telomerase. So this enzyme, what it does, it will maintain the length of a particular telomere. So whenever the telomere length is maintained, the cell undergoes replicative immortality. So cell can undergo as many proliferations. In normal cell, you can see 50 to 70 cell division, the cell replicative activity is lost and the cell undergo apoptosis. But whenever there is a telomerase reactivation, it can goes for as number of proliferation, and this is one of the hallmark which the cell cancer cell try to acquire, and it can be done with the help of TERT gene, the TERT specifically the TERT promoter region. So the TERT promoter region gets mutated, and you have activation of this particular telomerase, and this is this hallmark is acquired predominantly in the CNS tumor like gliomas. And the next is squamous cell carcinoma skin, and few of the cases of liposarcoma and prostate carcinoma also the TERT promoter gene mutation is seen. So this is the first important function of maintaining the telomere length. The other supporting role which TERT also has, it will modify the function of signal transduction pathway. It will regulate the gene. It will also have role in mitochondrial metabolism. So apart from maintaining telomere, it will have supporting role of other functioning which give the tumor cancer cell an edge to proliferate or to replicate n number of times. So this is an important hallmark where the third gene has a role to play and seen in commonly in gliomas or CNS tumor. Now we'll discuss the emerging hallmark, which is associated with the genomic instability or DNA repair pathway. So there is a lot of insult which occurs to a DNA, either due to the mutation or reactive oxygen species or radiation. Okay. In response to this insult, the DNA, as we see, P53 arrests the cell cycle, and it will give chance for DNA to repair its damage. Okay. If the damage is not good, it will further causes apoptosis and the cell is died. Or if there is a major DNA repair or minor DNA repair and the cells again proliferate. Okay, so anything which the genes which are taking part in DNA repair get altered, so you can get cancer because you don't repair the DNA and there is a proliferation of that broken or that unstable DNA strand, and you get cancer. A very specific carcinomas are associated with this pathway. One is breast carcinoma, ovarian carcinoma. Now the prostate and pancreatic also have a role where DNA repair pathway is involved. So DNA repair pathway, there are two subdivisions. One is where the single stranded DNA damage occurs and where double stranded DNA damage occurs. Single damage whenever there is only single nucleotide. So you have base excision repair where the base is excised and it is repaired by its complementary nucleotide. Then you have nucleotide excision repair where one to two means more than one nucleotide of a single strand is damaged. So it will try to repair same as base excision repair. Then you have mismatch repair. We have seen in colorectal where the mismatch repair genes get altered and you get HNPCC. Similar, there can be mismatch base repair. Okay, so mismatch repair genes have to function so that whatever the wrong nucleotide which gets attached to a particular repairing DNA strand has to check, and you have to add the correct nucleotide so that the strand gets repaired completely. So this single strand DNA damage doesn't have any robust checking mechanism where a DNA damage can be seen robustly. There can be a wrong base excision also. Okay, so if there is a wrong base excision repair also occurring in DNA repair pathway, it will further go to the apoptosis pathway as TP53 can detect. It is a wrong repair which has occurred. But when you come to double stranded, you have a homologous recombinant repair or non-homologous. So homologous means the repair of a particular strand of DNA is done with the help of its homologous strand. Okay, and this is a very robust repair pathway. It has a proofreading mechanism also, and majority of the large repair pathway, large repair in DNA occurs due to this homologous recombination repair. Same way, non-homologous, the homologous strand is not involved. It is just the end joining of a particular strand which is occurring. So these are the broad classification of the DNA repair pathway, of which the HRR is having a significant importance in solid carcinomas, and there are targeted therapies which are present against the homologous recombinant repair. When we see the frequency of malignancies which have this type of alterations, we can see endometrial carcinomas. We have arid one, which is present, which we have seen is associated with switch pathways. We can also have ATM, which is a HRR gene. So homologous recombinant gene one is ATM, is associated with endometrial carcinoma. We have in red color are the BRCA alterations, which we can see in ovarian. It is in great frequency. Similar in breast also we have alteration in BRCA mutation, BRCA gene. So there are a lot of malignancies where the DNA repair is involved, but the focus primarily will be the targeted therapy primarily under trial is on breast, ovarian, prostate carcinomas, and pancreas carcinomas. So what is the targeted therapy or targeted approach undergoing for this particular repair pathway? So there is one enzyme called poly ADP ribose polymerase. This is a nuclear enzyme and it has a crucial role in all the DNA repair pathway. Okay, it is not associated with homologous. It is involved in all the repair pathway which we have discussed, the single strand as well as double strand. So whenever there is normal PARP dependent repair and homologous functioning also, then cell survive, cell repair it proper DNA, and the cell survive and proliferate. Then there are scenario where the PARP is functioning but homologous is not, homologous recombinant is defective, but PARP is there, so it will try to shift it repair pathway from this homologous to the non-homologous, and it will try to repair, and there will be cell survival. So this particular both the strategies, PARP dependent repair and homologous, is taken into consideration. So we have to check whether homologous recombinant repair pathway is altered first, because if there is an alteration of this homologous recombinant repair, so there will be no repair occurring of a particular gene, and the cells which have this alteration, we can give this PARP dependent or PARP inhibitor. So the PARP inhibitor will prevent the function of this repair gene. So there will be no other repair mechanism which can be taken by this PARP gene. So in both the case, there will be no repair and cell undergo apoptosis. So this common strategies is been carried out or is under trial for breast carcinomas, ovarian carcinomas, pancreatic and prostate. It is still under trial. So knowing this repair pathway is also important path of solid tumor. So breast carcinoma, we know one of the pathway is this HRR pathway or repair pathway. The other alteration what we can see is, we have already discussed signal transduction PIK3CA. There is one gene ESR1 mutation. So ESR1 will encode for a particular receptor where estrogen goes and bind. So estrogen receptor alpha is encoded by this gene. So whenever you have this mutation of ESR1, your aromatase inhibitors or tamoxifen will not function. It will have a resistance, and this particular mutation is also now taken as a follow-up in a follow-up strategy, where the ESR mutation can be detected on a ctDNA, and ESR mutation is now gaining importance in breast carcinomas which are ER/PR positive and HER2 negative. We have ovarian carcinoma. So ovarian carcinoma can have, we know Lynch syndrome. We have already discussed MMR or mismatch repair mechanism is one of the pathway. Whenever it is hereditary acquired mutation, so you get Lynch syndromes. So you have either clear cell or endometrioid type of carcinoma whenever you have MMR pathway involved. You can have BRCA1, BRCA2 mutation where it is associated with high-grade serous carcinoma. You can get acquired mutation in TP53 also in such cases. So there is a molecular substratification of ovarian carcinoma. Whenever you have BRCA1, BRCA2 mutation, you can try you can go for a PARP therapy in such high-grade serous carcinoma. You have other carcinoma like endometrioid carcinoma, where alteration in genes like poly. So poly genes are basically a polymerase gene which is having proofreading activity, and whenever the poly or pol D mutation occurs, there are a lot of the cell actually loses the activity of its proofreading, and you get lot of acquired mutation in particular gene. But this poly mutation is associated with a good prognosis. It is a hypermutated condition, but this is considered as a good prognosis in case of endometrioid carcinoma. You have other low grade and mucinous which is associated with RTK-RAS pathway, and you can have CDK into cyclin dependent kinase inhibitor alteration also. So all the major category of ovarian carcinoma has some molecular alterations which are different from each of the category. So target intervention, we already know, knowing the particular mutation in breast ovarian carcinoma, if there is DNA repair pathway alteration, we have PARP inhibitor. If there is PI3K pathway, we have PI3K inhibitor. So therapeutically also, knowing mutation in breast and ovarian carcinoma carries significance in molecular technique. In molecular technique also, we have viral induced oncogenesis which we detect by molecular technique, where HPV has a role, where HPV induced cervical cancers, in which we can check which type of HPV 16, 18 or other non-harmful HPV is present in a particular tumor sample. So HPV has two component, E6 and E7. E6 will cause TERT promoter mutation. It will suppress the P53. Similarly, E7 is a component which goes and release this RB from E2F. So E2F is a transcription factor, and once transcription factor is released, it leads to proliferation. So this is a basic mechanism of HPV induced squamous cell carcinoma, and this HPV sub classification can be detected by molecular technique. Similarly, we have EBV, which can be detected by IHC also, as well as you can check the RNA expression of a particular EBV gene. So molecular technique can also be implemented for EBV alterations, and this commonly seen in hematolymphoid B cell lymphomas, and more towards solid we have HPV. Now, in brief, we will discuss what, after knowing what are the alteration we see in the solid tumor, what are the test assays, or what are the testing platforms, or what are the available techniques present in the molecular diagnostic to detect all these translocation, mutation, or methylation. So we can detect individually single test by PCR, or we can sequence a particular DNA, and we can just detect the mutations, whether it is deletion, insertion, or we can use the MLPA whenever we see large scale transition, whenever the large part of chromosome is deleted or inserted, that can be detected by molecular platform like MLPA, or you can go for cytogenetic techniques where you can detect such large scale transition. Then we have next generation sequencing, which encompasses all of this translocation, mutation, CNV, methylation. It provides advantage of massive parallel high throughput sequencing. You can get all the type of alterations. You can go for entire genome sequencing, exome sequencing, and you can also do a single cell sequencing where you just focus on a single cancer cell and see whatever are the alterations. You can also use this technique for knowing the epigenetic changes like methylation. You can study the protein structures or protein alterations with next generation sequencing also. You can check the gene expression profiling with the help of next generation sequencing, and non-coding or microRNA study can also be done with next generation. So next generation sequencing provide a comprehensive approach in molecular lab in cases of solid tumor as well as the hematolymphoid malignancies. So these are the molecular platforms or machines, PCR, Sanger sequencing, and next generation sequencing, which are routinely used in our molecular diagnostic labs. So PCR is a basic workforce of any molecular lab. So PCR is basically polymerase chain reaction, where we will try to amplify our DNA of interest by adding primers, which goes and bind to a particular specific region in 5' end and 3' end. We will put the DNA primers, we will put the enzyme, Taq polymerase is the enzyme, we will put the DNTPs. So there will be breakage of there will be, first step will be the breakage of two-stranded DNA, then the primer will go and bind, and the Taq polymerase will lead to the formation of a new strand of DNA with the help of DNTPs and other component of a PCR reaction, and with each PCR reaction you can get 2 copies of DNA. So this cycle undergoes 20 to 40 times, and for all the molecular tests, the first step is PCR. We have to amplify a region of interest, and then we check on the downstream other pathways like Sanger, next generation sequencing, real-time PCR, or conventional PCR. So in PCR, there are three phases: exponential phase, linear phase, and plateau phase. So once this PCR reaction starts in PCR thermal cycler, you have some reaction which is occurring in the background, but there will be time comes where it will cross this threshold, and this is called CT value or cycle threshold. Once the cycle threshold is crossed, you get exponential phase, where with each cycle you are doubling the number of your DNA strand. Then linear phase, the reaction will slow, and plateau phase, you will just stop the reaction. There will be no doubling or there will be no amplification of our DNA strand occurs. So there are two type of PCR routinely done used in our molecular lab. One is a conventional PCR, where you amplify a particular DNA, and at the end of the PCR you will visualize that particular DNA, whether that mutation is present or not. So this is the area where at the end of the PCR you will get the PCR product. Then there is a real-time PCR. With each nucleotide sequence added, there is a fluorescent signal release, and you count that particular real time, and you just quantify the copy numbers, how much that particular copy numbers are present in a particular PCR reaction. So these are the two PCR which is routinely used in the diagnostic molecular diagnostic lab. So conventional PCR, we use this gel doc system to visualize in gel electrophoresis what are PCR product we get at the end. Then real-time PCR uses few dyes, like there is cyber green dye and there are probes. So what are the difference between both the techniques? Cyber greens are the dye which are the intercalating dye. They go and bind to the double stranded helix of a DNA and it fluoresces. So they will just calculate the bounded and unbounded cyber green and they will give you the copy number. But the only drawback is in a single strand there can be more than 2 or 3 dyes can be getting bound. So there can be false positive increase in quantification or number of your PCR product. Then we have probe. So probe is nothing but a sequence of nucleotide. We have two fluorescent tagged dye, reporter dye and quencher dye. So it works on a principle of FRET. So what is the principle? Reporter and quencher dye remain in close proximity so that this reporter fluorescence is just remain in check whenever they are in close proximity. So whenever quencher is near reporter, this reporter fluorescence will not be detected. So whenever a PCR reaction from 5' start, this tag polymerase will add the nucleotide and it will come to the reporter. And once it comes to the reporter, this tag polymer has an exonuclease activity, so it will cut this reporter. So once the reporter is get cut from the quencher, it gets away from the quencher dye and it will give a fluorescent signal. Okay, so the fluorescent signals start to be detected in reported dye once a strand is completely formed, or there is a the PCR reaction has been completed. Okay, so this is a basic difference between the two routinely used fluorescent dye. Okay, then we have newer type of PCR reaction which is called digital PCR. So what is the difference between this digital PCR and the real-time PCR? This also has a fluorescent dye, but the PCR reaction in digital PCR is done in a compartment. So the PCR, whatever PCR reaction which we have put in a tube is first compartmentalized. There will be a compartment in which this particular PCR reaction will go, and in each compartment there will be amplification of a particular strand of DNA. So with each amplification you will get a fluorescent signal because this particular are tagged by fluorescent dyes, and this fluorescent dye can be in various colors. So we can have wild type have particular red color fluorescent dye, mutant can be have red color fluorescent dye, and you can actually count the number of mutant copies as well as wild type copies in cases of solid tumor. So I will give you one example of EGFR. EGFR will have a wild type copies also as well as mutant copies also. So to get the information of what is the mutant copies of EGFR, you can tag a particular with a fluorescent dye. The mutant copy will bind to mutant region of EGFR. Wild type will go and bind to wild type. There will be a PCR reaction which will take place in particular compartment that will get counted as the cell as this particular compartment get fluorescent signal, and we can get these are the two mutant copies. There is one double positive which we don't have to count, and there is a green wild type. So you can just quantify this particular mutant gene copies. The advantage is that this particular technology is now taken into consideration whenever we have to have follow-up of a particular malignancies to know the residual disease present after the treatment, and it is having a sensitivity of 10⁻⁴ to 10⁻⁶, and this particular technology is also is having a reduced susceptibility to PCR inhibition, which the real-time PCR doesn't have. And real-time PCR, you will require a standard sample where we compare the copy numbers with the standard amplification. So these are all the advantage of digital PCR, and it is coming up as in solid tumors also, in liquid tumors also. In both the places, it is having a role in residual malignancy detection. Then we move to the next Sanger sequencing. It is a first generation sequencing. It is a gold standard technique as far as sequencing is concerned. So this particular sequencing technique is based on the dideoxynucleotide. So what is the dideoxynucleotide? So you have DNTPs with an hydroxyl end. So whenever there is a one less hydroxyl group in a particular DNTP, it is called dideoxynucleotide. So this hydroxyl group at the second position is required by the upcoming nucleotide whenever there is an extension of a particular DNA strand. So whenever new nucleotide has to come and bind to this nucleotide, it will require that hydroxyl group for the attachment, where the phosphate of the other nucleotide bind to the hydroxyl group. But whenever one hydroxyl is not there, so the PCR reaction or the sequencing get terminated there. And this principle, along with the fluorescent tagged dideoxynucleotide, is utilized in Sanger sequencing. So what happens in a tube? You have PCR reactions, you have dideoxynucleotide as well as the normal nucleotides also. So they will go and start forming a new strand from 5' to 3' end, and there will be sequences where this dideoxynucleotide go and bind, and that particular DNA strand chain elongation is terminated. Okay, so it is called chain termination reaction also, and with each termination there is fluorescent signals which is released. So there can be a lot of different sizes of DNA fragment which we can see in a particular tube. These different sizes of DNA fragment then undergoes the capillary gel electrophoresis, where it will separate the fragments of DNA based on size, and you get this kind of electropherogram. Okay, where you have the nucleotide on the top, and you have the peak of fluorescent signals in a form of this graph. So whenever we finish our Sanger sequencing, we get such type of sequence, and we compare it with the normal human genome, and we read this manually, all the sequences, and we try to find whatever alteration, comparing it with the normal human reference genome. So if you are reading KRAS exon 2 mutation, so you should have normal KRAS human reference genome. You compare these particular sequences with the sequences of the normal human reference, and you see whether there is a change in particular nucleotide, missense mutation, there is insertion mutation, which you can also detect by the Sanger sequencing. The only downside is it is very labor intensive. You cannot go for higher multiplexing. The longer gene like BRCA1 or BRCA2, which are more than size of 5 kbs, cannot be done in a single Sanger sequencing reaction. So these are all the disadvantages of particular Sanger sequencing. So to overcome the Sanger sequencing, there are a lot of other second generation sequencing like pyro sequencing was available. Then the next generation sequencing came into picture, which is based on the sequencing technique, but it will give an advantage of doing a lot of genes, high throughput, and you can check mutations, you can check deletions, you can also check the epigenetic alterations. So these are the two techniques routinely used in molecular lab. For colorectal, we have sequencing assays for KRAS, NRAS, BRAF, TP53. For breast, we have BRCA1, BRCA2 can be done in Sanger sequencing after the modification of Sanger sequencing technique. There are PCR, real-time PCR and digital PCR available for single mutation for EGFR, PIK3CA. So these all PCR reaction can be done for a single particular gene. You cannot do large scale of multiplexing with this PCR assays or Sanger sequencing. But whenever you have to go and see a particular mutation, you know this particular alteration is commonly seen in particular carcinomas, you can go and check for the particular mutation by single assay, because these are cheaper than the other next generation sequencing assays. So this also carries importance whenever you have a specific alteration to look for in a carcinoma cells. Third question is fluorescently tagged dideoxynucleotide are used in which type of PCR molecular technique? Real-time PCR, digital PCR, Sanger sequencing, or conventional reverse transcriptase PCR?
Um, Sir, all the participants are answering the option C,
Yes.
Sanger sequence.
So we have already discussed this dideoxy technique causes chain termination in Sanger sequencing, and which is an essential component of our Sanger sequencing assay. Now we have next generation sequencing, which encompasses large part of molecular genetics. So after extraction, there is a step of library preparation. So this library preparation is nothing but the particular DNA is fragmented or broken into pieces, and it is having one adapter which is attached to a particular DNA strand or cDNA strand whenever we are going for a RNA sequencing. This after adapter attachment or ligation, this goes in a sequencing machines. So there are two commonly used platform, Ion Torrent and Illumina. And after sequencing, you get a lot of sequencing reads or the sequencing information, which is in the form of FASTQ files. These FASTQ files are random. Now, to make sense of this particular sequencing, whatever we have done, we should have something like filter or something so that we can get the information about a particular mutation. Okay, so for that we have bioinformatic analytic pipelines. So bioinformatics is basically informations which this particular biological sample has given. So this particular field deals with getting the information from the sequencing. So these are nothing but filters which we are using, but these are little bit complicated filters where the arrangements are occurring. Then we analyze that particular arrangement and we make out this is a pathogenic variant or this is a benign variant. So this FASTQ files first get converted into BAM file, which is called binary arrangement of a particular file, where the files are arranged in a particular read or particular family. And once these are arranged, we just align to a normal human genome and we just convert into VCF files. Similar to the Sanger sequencing, where we are reading the manually whatever after comparing to the normal reference genome. Here, everything is done by the system or the software, which at the end will give you all the variants available. And after we receive all the variants, we have to find whether that is pathogenic, benign, or it is doesn't have any significance. And then for the pathogenic and likely pathogenic variant, then we have to report in next generation sequencing. So next generation sequencing, I have already discussed what are the advantages. It will give epigenetic information. It is having high throughput and multiplexing. No prior knowledge of sequence required for Sanger and PCR. We should have a prior knowledge of sequence to design the primers, but in case of next generation sequencing, that is not required. So these are the these are the panels which are routinely used in molecular lab. All the red color genes are what we have discussed in this lectures, all the transduction pathway to all the other hallmarks. So these are all covered, and apart from the discussed one, there are other alterations also which we can detect after the next generation sequencing. You have HRR pathway where we have only spoken of BRCA1, BRCA2. There are other alterations also in HRR gene pathway. So in NGS, you get comprehensive information in a one go or one assay. Okay, in Sanger and PCR, you have to go one by one for all the assays. So eventually it will take more time when you compare to check for mutation in more than 1 to 2 genes. Whereas in next generation sequencing, you get a lot of gene information in one sequencing assay. To summarize, I have already spoken about the hallmarks of cancers where the genetic alteration, transaction pathway, uh, TERT mutations are there. We have discussed about few of the inhibitors which are associated with the hallmark of cancer, and this molecular diagnostics help just aid in the diagnosis of few of the solid tumors. It provide a prognostic insight of a particular solid tumors. For example, TP53 mutation, we can get information about the therapeutic approaches. Like for EGFR mutation, we know TKI is there. For KIT mutation, after the molecular diagnostic, we can give a specific TKI inhibitors. And research and development, this is an important tool. So more and more information can be gathered after doing the molecular analysis of a particular cancer cells, and we get more and more biomarkers, more mutations as we move forward. So thank you. This was a very long talk, uh, but I tried to cover as many as possible as far as the solid tumor is concerned. So I will ask Dr. Kumar Prabash sir to just conclude with the remarks.
Prabhas sir, you are muted.
Yeah. So, good evening, everyone. And this was a nice lecture with a lot of overview regarding what we do in diagnostic in pathology and especially molecular pathology. Uh, Over the time period, looks like we require for individual tumor a separate lecture. One lecture covering everything is tough in the time. But what I'll do is I'll put a clinical context to it.
Yes, sir.
A small example in the tumor which I deal with it. So tumor which I deal with, those tumor which has a very mystic outcome, and this is one of the tumor is that is lung cancer. So if you would have asked me around 10 to 12 years back, and there were some examples shown, patients were treated like all lung cancer was cancer. People wanted to say, hey, you divide between small cell, non-small cell. For a clinician, it doesn't matter because treatment was same. Platinum used to give for non-small cells to give a small cell. But time went by small cell and non-small cell. In non-small cell, we wanted to know squamous, non-squamous. In non-squamous, we know at least buckets, and 10 buckets are shown in the good slides. That it is EGFR, it is ALK, ROS, MET, RET, KRAS, NTRK, PDL1. Then again, we want to know in PDL1 what subgroups are there. And this is slowly and steadily happening to many tumors. This is not only restricted to lung cancer. And did it impact the outcome? It looks like that, for example, and this was time when I was getting trained. The average life of the lung cancer patient used to be 6 to 8 months. Now, the average life of these patients which has mutation is around 40 to 50 months. ALK positive patient, today 5-year survival is 65% if you can give state-of-art treatment to them. So that is the kind of change it has made in lung cancer. In many other cancer, it has had a different and differential impacts, but looks like we are not going to get it are going to have these molecular markers which are going to stay with us, and which we need to handle as time goes by. And yes, slowly and steadily, it has a definite impact on many tumors, and people are evaluating many of those that are we able to get this molecular test done or not. And the reason being because it changes the outcome of treatment based on giving treatment further, that targeted therapy, what we call, for that matter, immunotherapy, what we call, or defining the prognosis and then intensifying and de-intensifying treatment. So I enjoyed the lecture.
Thank you, sir.
Hopefully my input is useful for the students who are listening, that what they are doing and what they're going to do have a clinical bearing when the report comes.
Yes.
And what kind of report comes becomes important. That is a part of technology. I'm sure that technology part, sure you and your colleagues are far better off to understand it, how to ensure that the right report comes to the clinician in the clinic. Thank you so much for inviting me. I enjoyed the lecture.
Thank you, sir.
And if there's any question, I'll be happy to take.
So we have few questions.
Yeah.
One question is, are the epigenetic alterations targeted by any therapy?
Yes, we can have therapies which can target particular epigenetic. So we have methylation inhibitor therapies. We have HDAC, which causes deacetylation of a particular gene. So we have HDAC, we have hypomethylating agents, we can give in such, if we get epigenetic changes, we can give this particular inhibitors. But I think the role in all the solid tumors is not well established. So in well established only this kind of therapies can be tried. Sir can give a more insight on the therapeutic part, sir.
Can you repeat the question? Sorry, I missed
Yes,
that.
yes, sir. The question is, are the epigenetic alterations targetable by any therapy?
Yes, there are demethylating agents, which is available, like azacitidine, is there, venetoclax is there. So there are, drugs which is slowly and steadily getting into epigenetics and having an impact.
Another question is, what is canonical mutation?
Okay. So canonical is what that particular gene has to function normally, and non-canonical, which is not the usual the pathway with that gene will function. So the non-canonical is what I have shown you in the TERT. So it will have an additional function in metabolism of mitochondria or signal transduction, but the usual function of the TERT is maintaining the telomere length. So that is a canonical function, or you can say canonical mutation, which causes to maintain the telomere length by impacting the TERT gene.
Thank you, sir. Sir, someone is asking which book to refer for molecular pathology.
Difficult to difficult to tell which book to go for molecular genetic as per se, because this is so dynamic field na. Every month there is a new molecular alteration coming, and there is a lot of publications is happening in this field. So I think for basics molecular also you can go and search a good review article from a standard journals which is having good indexing, and you can go and read. It is very knowledgeable journals, can give basic knowledge as well as also the techniques or whatever the new alterations coming. And I think MDPI is one good journal which tells more about the basic of the molecular genetics. Rather than books, I will prefer to go for such publications.
Thank
But
you,
reading
sir.
the basic books for what are the DNA, what are RNA, I think pathology book will give that information.
Sir, someone is asking, in a peripheral setup, is it a must for NGS solid cancer?
Can I take this question?
Yes, sir.
Yes, sir. Sure, sir. This is for you only.
It's so when it is not feasible, we may skip it. But why not make it feasible? Why we want, is to say that one place, it is not required because it is a tier 2, tier 3 city. Today you have a central labs, and doing test at the cost which is far lesser than doing, for example, I'll give you a lung cancer. Depends upon cancer to cancer.
Thank you
But
very
I'll
much.
give you if you want to do ALK by IHC, ROS by FISH, and EGFR by PCR, the cost is the same will you get from NGS, and you will be doing those 15, 20 genes, where 10 of them will be have a treatment where you can give treatment to the patient. So you have a labs who are willing to go and get the sample place for things there. We need to try to do that. So yes, it is peripheral or not, it is required. Question is that, are we willing to understand how important it is for the patient and willing to put effort to make it reach to that place so that we get the report?
Thank you so much, sir.
Sir. Sir, Dr. Babita is asking, please elaborate microsatellite instability pathway again.
Okay. So, as I told you, microsatellite is basically a repeat of a particular nucleotides, and if it is occurring in a tandem, so whenever there are 6 tandem repeat of a particular nucleotide, it is called microsatellite. So this microsatellite always remains stable whenever there is a mismatch repair gene functioning normal. Okay, so whenever this mismatch repair gene functioning or is altered or mutated, this microsatellite stable become unstable, and this unstable occurs. Wow, you will get microsatellite repeats one after the other. And what this microsatellite repeat will do, it will have an effect on a cell proliferation or the genes which having cell proliferation activity. Okay, so the pathway which causes the activation of the cell proliferation pathway is not well understood. It is still under study, but this is what the microsatellite instability refers to. And it is also be in colorectal carcinoma specifically, this microsatellite instability pathway is very important, because immunotherapy to be given or not given also depend on the MSI high and MSI low, as we have seen in one of the slide, where the four consensus molecular subtyping was there. And the first, there was MSI high, where you can give the immunotherapy as a treatment. So knowing about this microsatellite instability by this mismatch repair gene is important.
Right, sir. Sir, thank you so much, Dr. Rakesh, for such a elaborative talk. And you have made this complex topic very simple with your beautiful presentation.
Thank you.
Also, I would like to thank our chairperson, Dr. Kumar Prabhas, for sharing their clinical insights into this field. Also, I would like to thank all the participants for their